Okay, we'll go ahead and get started. We're going to talk about the nervous system today, as we finished the endocrine system last week. Nervous system, we'll start with the central nervous system, the peripheral nervous system, and then some of the cells that are involved in making up the nervous system. As you know, the central nervous system is sort of the computer for the body, composed of a network of nervous tissue that transmits electrical impulses throughout the system to control the multiple functions that go on inside the body. The simplest response requires some level of cooperation amongst systems, and so when we look at the series of responses that are required for the integration of sensory information and the coordination of motor responses, the central nervous system is what poses, the nervous system as a whole, is what serves to provide this integration and coordination of systems. Simple automatic responses directed at a subconscious level followed by complex variable responses are under conscious control. All of these functions are performed by the nervous system. Now, we've already talked about the endocrine system, and we recognize that there are some components of the nervous system that are shared with the endocrine system as well. The endocrine system, remember, chemical substances are secreted inside the body. These chemical substances take a period of time to carry out their effect. And so the endocrine system is not as rapidly acting as we'll see the central nervous system or nervous system tissue. The nervous system provides swift but generally brief responses to stimuli through temporary modifications of activities at other organs, whereas the endocrine system shares many of the characteristics. The system is much more slower in inducing its response. The endocrine system adjusts metabolic operations along with other systems. It produces longer-term responses. The endocrine system coordinates growth and maturation, sexual development, those responses that we've already talked about. But when we compare the two systems, we see that the endocrine system, even though it induces its effects sometimes rapidly, as we talk about with the flight or fight type of response, we see an intermixing of the two systems. We see hormones and chemical substances produced by the endocrine system acting on the nervous system to carry out particular responses. And so we can see it can act comparatively for short periods of time, as with the fight or flight type of emergency. The endocrine responses typically are more slower than the nervous system, as I already alluded to. It's in part fact because this information, these commands, the chemical substances, have to be sent in chemical messengers in the form of hormones, distributed in the bloodstream. The bloodstream then takes it to the area for the reaction to take place. So this type of mechanism takes a much longer period of time than nervous system, which is sending an electrical stimulation that's going to induce a response. Nervous system includes all of the neural tissue of the body. The basic functional unit of the nervous system are cells referred to as neurons. There are supporting cells in this system as well, neuroglia. These are separate. They are products of the neurons. They provide a supportive framework of neural tissue. They act basically as phagocytes, and if you remember in anatomy and in microbiology, we talk about phagocytic activity, phagocytes, which help regulate the composition of interstitial fluids. But phagocytes, when we talk about in other systems, these are typically cells that will come in and they're scavengers. They pick up cellular debris. They can kind of rid systems of foreign invaders. They help destroy other cells that may be foreign to our body. We'll see a significant network of phagocytic cells involved as the supporting cells in the nervous system. Neuroglia are called glia cells, far outnumber the neurons and account for roughly half the volume in the nervous system. Two major anatomical divisions in the nervous system, the central nervous system and the peripheral nervous system. In the central nervous system, this consists of the spinal cord and the brain. Anatomical structures you need to be familiar with from the lab portions of the course. But so central nervous system consists of spinal cord and brain. These are complex organs, include not only neural tissue but also blood vessels and various connective tissue that provide physical connection and support. The central nervous system is responsible for integrating, processing, coordinating sensory data and motor commands. Sensory data conveys information about conditions inside and outside the body. Motor commands control and adjust activities in peripheral organs and in skeletal structures. Now also keep in mind, sensory information is important for us almost at an unconscious level. When we walk, it's the sensory information that the nerves that sense where our feet are on the floor sends a signal back to the brain to help us maintain our balance and help us to be able to take the next coordinated step. So this has to be an integrated system of both sending out a motor signal to initiate a voluntary response by the muscles in our legs, but also it has to be a sensory input coming from as the tactile stimulation of our foot strikes the floor, telling us what the position of our body is and where the next step needs to be. And this happens instantaneously. So when you look at the effect of the nervous system, especially when you compare the nervous system to what the endocrine system is, we discussed last couple of weeks, you can see where the nervous system is instantaneous responses versus an endocrine system which is a little more slower in taking place. The central nervous system integrates information regarding your balance, position of your limbs, coordinates, helps you to recover, sending motor commands to appropriate skeletal muscles, all within split seconds. Also, central nervous system, particularly the brain, is also the seat of higher functions such as intelligence, memory, learning, and emotions. The peripheral nervous system, on the other hand, includes all the neural tissue outside the central nervous system. Peripheral nervous system delivers sensory information to the central nervous system and carries motor commands to peripheral tissue and systems. Bundles of axons or nerve fibers, carry sensory information and motor commands to the peripheral nervous system. These particular bundles are associated with blood vessels, connective tissue, are called peripheral nerves or simply nerves. Nerves connected to the brain are called cranial nerves. Those attached to the spinal cord are called spinal nerves. The afferent division of the peripheral nervous system, the afferent division, focus on the word afferent division of the peripheral nervous system, brings sensory information to the central nervous system from receptors in peripheral tissues or organs. Receptors are basically sensory structures, either that detect changes in the internal environment or they respond to specific stimuli. And these structures can range from dendrites. Dendrites, as you recall from nervous tissue that you studied in AMP1, dendrites are components of the nerve cells. We see receptors that may be on neurons or specialized cells of other tissue. Merkel cells in the epidermis. If you remember in the integumentary system, we talked about the Merkel cells that help to in the tactile stimulation of different surfaces in the hand. Those little cells help us to sense better in the little pads of our fingertips to relay sensory information. The efferent division of the peripheral nervous system carries motor commands to the central nervous system from the central nervous system to muscles and glands. So efferent division, peripheral nervous system, carries motor commands from the central nervous system to muscles and glands. These target organs which respond to doing something are called effectors. The efferent division is mostly, or has both somatic and autonomic divisions, which we'll talk about in much more detail throughout this lecture. So when we look at the somatic nervous system, the somatic nervous system controls the skeletal muscle contractions. Voluntary contractions are under conscious control. When we think about striated muscle, skeletal muscle, we think about voluntary control. These are the muscles we have conscious control over. Most of the smooth muscles, the non-striated muscles, we do not have conscious control over. We cannot determine the peristalsis movement of our esophagus and our stomach, the wave-like contractions that our stomach and our bowel and our intestines will do. That happens automatically. We cannot control the movement of the ducts that lead from the pancreas. The smooth muscles inside those ducts will squeeze and bring digestive fluid into our stomach. We can't control the smooth muscle movement of blood vessels. We have no voluntary control of the muscles relaxing and contracting in our circulatory system. Those are smooth muscles. We do not have conscious control. but when it comes to flexing our forearm, extending our forearm, scratching our back, the movements of those skeletal muscles, those striated muscles, we have voluntary control over. So in the somatic nervous system, which controls skeletal muscle contractions, voluntary contractions are under conscious control. Involuntary contractions may be simple automatic responses or autonomic responses to movement or complex movements. They are controlled at a subconscious level and outside our conscious awareness. Accidentally placing our hand on a stove causes a response in which we withdraw our hand away really quickly. We don't have to think about that process. Now that does involve skeletal muscles, but remember that's an involuntary contraction based upon a painful stimuli. And so it's a response. It's outside of our conscious awareness, but it still involves striated muscle. So reflex movements in the somatic system are also possible, but again, this is primarily a system that involves conscious control. Now, the autonomic nervous system, which we'll talk about, I don't know if we'll get into it today, but certainly next week. The autonomic nervous system, or the visceral motor system, provides automatic regulation of smooth muscles, cardiac muscle, glandular secretion. This is at a subconscious level. The autonomic nervous system includes two divisions. That's the sympathetic and the parasympathetic division. This is commonly, when we look at these two systems, they have an antagonistic type of a relationship. If the sympathetic nervous system speeds up the heart, the parasympathetic nervous system slows it down. If the parasympathetic nervous system increases GI motility, gastrointestinal motility, the sympathetic nervous system slows it down. So they have an antagonistic relationship. And so as we see, the two systems will complement each other. If we need more skeletal muscle, if we need a fight or flight response, then more blood flow to the skeletal muscle, more activity to the skeletal muscle occurs, then we'll see greater stimulation. We'll see greater stimulation of the sympathetic nervous system. If we want to digest food, then we see a slowing of sympathetic stimulation to skeletal muscle. We see an increase in parasympathetic stimulation to skeletal muscle, which slows down the skeletal aspect, and we see an increase in parasympathetic stimulation to the gastrointestinal tract, and a decrease in sympathetic stimulation, which would increase bowel motility. So the two systems will work in opposite. The cell body, or the soma, this contains a relatively large round nucleus and a prominent nucleolus. The dendrites, variable numbers of slender, sensitive processes known as dendrites extend out from the cell body. Typical dendrites are highly branched. Each branch bears a fine process called a dendritic spine. In the central nervous system, neurons receive information from other neurons, primarily at the dendritic spine. This represents about 80 to 90% of the neuron's total surface area. The axon is the long cytoplasmic process that's capable of promulgating electrical impulses. Again, going back to AMP1, when you had to identify the nerve cell, you saw the body of the nerve, you saw the dendritic processes, you saw the long axonic process that led from the main body of the nerve. Now, just as we see cytoplasm inside the cell, we talk about nervous tissue, we talk about axoplasm. This is the cytoplasm of the axon. It contains neurofibrils, neurotubules, vesicles, lysosomes, mitochondria, and various enzymes. Just as we see in a regular cell, the axoplasm is surrounded by an axolema, which is a specialized portion of the cell membrane. In the central nervous system, the axolema may be exposed to interstitial fluid or covered by a process of neuroglia. The axoplasm is surrounded by the axolema. This is a specialized portion of the cell membrane. The telendria, this is the main axon trunk, any collateral ends in a series of extensions. The telendria is basically the end of the dendron. There's also the synapse, which you, again, may be familiar with from the muscular system in AMP1. This is the part of the synapse, specialized site where neurons will communicate with another cell. Now, neurons, as we'll talk about, can communicate with other neurons. Neurons can communicate directly with muscle. Typically, there's a chemical messenger or neurotransmitter involved in this process that's released at the synaptic terminal that will carry out a particular type of an event or will stimulate the muscle to react in a prescribed way. The synapse, we have two cells that will meet. There is a presynaptic cell, which has a synaptic terminal that sends the message. Then there is the postsynaptic cell. This is the cell that is after the point of where the two cells will meet. This receives messages. This is the portion of, or the cell that will receive the message. If two neurons are meeting, one has a presynaptic terminal that will release a chemical messenger. That messenger will relay that information to the postsynaptic, or the second cell. So it's a point of communication, one presynaptic, one postsynaptic. The synapse is the gap or the space or the union where these two different cells will meet. This is illustration out of your textbook that basically shows a synapse. Here you have two nervous neurons that are meeting, the body, the axonal process, the teledendria. Here is the point of synapsis. This is the presynaptic terminal on the first cell. This is the postsynaptic area on the second cell. So presynaptic, postsynaptic. Again, the structures inside the cell are those what you're similar to seeing before. You have a nucleus and a nucleolus. You have Golgi apparatus. You have both mitochondria, which are significantly packed inside these cells for energy. And so the intracellular organelles are similar to what we've seen previously. At the synaptic cleft, you see both an illustration and an electron microscopy where you can see, again, presynaptic and postsynaptic structures here. Presynaptic chemical messengers located here are about to be released at this cleft, at this opening, the space between the postsynaptic cell. Mitochondria obviously being a part because this process requires energy. And you'll see the release of these chemical messengers sending the message to the second cell, which is at the postsynaptic or the postsynaptic membrane of that cell. And that's it, an electron microscopy. Now the chemical messengers. We've talked about acetylcholine in AMP1, and we'll talk about some other neurotransmitters later on in this lecture series. Neurotransmitters are typically packaged in the synaptic vesicles. Remember, neurotransmitters are basically chemical messengers. Acetylcholine is the one that we have primarily been talking about up until now. In the presynaptic cell, this is usually a neuron. In the postsynaptic cell, it can either be a neuron or another type of cell. We've used the example of muscle cell before. When the neuron communicates with another, the synapse may occur on the dendrite, on the cell body, or along the length of the axon receiving the cell. A neuromuscular junction, a definition you're probably already familiar with, a synapse between a neuron and a muscle cell. The synaptic knob is a relatively simple, rounded area where the postsynaptic cell connects with another neuron or another type of cell. The term synaptic knob is widely recognized and will be used again throughout your textbook. The same structures are also called terminal buttons or terminal boutons, end bulbs, or neuropods. A synapse is a narrow synaptic cleft. It separates the presynaptic membrane, as you saw in the earlier illustration. It's where the neurotransmitter is released from the postsynaptic membrane, which bears the receptors of the neurotransmitter. The synaptic terminal at the neuromuscular junction is a similar process, but it's a little more complicated. And again, going back one semester in AMP1, we looked at that process at the neuromuscular junction. We talk about transporter materials between cell bodies and synaptic knobs. This is called axoplasmic transport. Axoplasmic transport. Remember, the axoplasma is like cytoplasm, and remember, cytoplasm is a dynamic fluid medium that's inside the cell. We also see dynamic movement of this axoplasm inside nervous tissue as well. Some materials travel slowly at a rate of a few millimeters per day. The transport mechanism is known as the slow stream. Vesicles contain neurotransmitters. move a little more rapidly. They're in what's referred to as the fast stream. And axoplasmic transport occurs in both directions. The flow of material from the cell body to the synaptic knob is referred to as anterograde flow. At the same time, we'll see other substances are being transported toward the cell body in what's referred to as retrograde flow. Now, let's throw a disease condition into this process. The disease condition that we'll talk about now is called rabies, the rabies virus. This is perhaps one of the most dramatic examples of clinical condition that relates to retrograde flow. Retrograde flow. The bite of a rabid animal, an animal that's infected with the rabies virus. It is the saliva or the secretions from that animal get injected into the tissue. In peripheral tissue, the virus particles quickly will enter into the body and enter into the nervous tissue at the point of the synaptic knob, at the peripheral axon. And then by retrograde flow, it then carries the virus back to the central nervous system. And it's a fatal condition. It causes a mass reaction. It's almost like a super antigen where it causes a massive inflammatory response from the body, leading to neurological dysfunction and eventually death. Now, there are some other toxins, including heavy metals, and some other pathogenic bacteria and other viruses that also can bypass the central nervous system defense by utilizing this axoplasmic transport process. Here's some different types of neurons. And remember, we said neurons can communicate with other types of tissue, like the muscle tissue. They also can communicate with themselves. The structures are variant in how these different neurons can appear. This is an anaxonic neuron, a bipolar neuron. You can also see the unipolar neuron. Again, notice the axonal processes that lead from here, but all of them come back to a centrally located body and nuclei, and then the multipolar neuron as it exists here. The anaxonic neurons are basically small. They have no anatomical features that distinguish the dendrites from the axons. All of the cell processes look alike. We see them located in brain and other special sense organs. Bipolar neurons have two distinct processes. One dendritic process branches extensively at its distal tip and one axon. Then we also, it communicates with the cell body between them. The bipolar neuron, as the illustration shows, it occurs in special sense organs where there's a relay of information. Sight, smell, hearing, receptor cells, and neurons. Bipolar neurons are smaller than the unipolar or multipolar neurons. Largest measures about 30 millimeters in size from end to end. And then the unipolar neurons, or pseudo-unipolar neurons, dendrites and axons are continuous. They're fused into the cell, and the body lies from one side or off to one side or the other. The initial segment lies where the dendrites converge. The rest of the process carries action potentials and is usually considered to be an axon. Most sensory neurons and peripheral nervous system neurons are unipolar. And then the multipolar neurons have two or more dendrites on a single axon. Multipolar neurons are the most common type of neuron in the central nervous system. The motor neurons that control skeletal muscles are basically multipolar. The axons of multipolar neurons can be as long as those of the unipolar neuron. The longest carries motor commands to small muscles that move the toes. And now you can see the two divisions, the central nervous system and the peripheral nervous system. And remember, afferent fibers, sensory neurons in the peripheral ganglia, and then efferent fibers under somatic motor neuron control. Then there are also visceral neurons, which will control the smooth muscles that we usually see in, again, blood vessels. We see those smooth muscles in tissue such as the gastrointestinal tissue. We see smooth muscles in glands. As you see, visceral motor control also includes cardiac muscle. Now, cardiac muscle is striated muscle, but recognize cardiac muscle is not under voluntary control, and it's the exception to the striated muscle we talked about under voluntary control earlier, and some affect adipose tissue or fat tissue as well. Now, sensory neurons and the somatic sensory neuron motor outside the world are positioned within it. It's important to be able to monitor the circumstances that go on in our environment so we can manipulate the body, make conscious and or subconscious responses. The different effects on our body are made by the environment which we're on. We get exposed to a cold temperature, then an automatic response in the body is to contract the pores in our skin. And so our skin, the smooth muscle will immediately contract. The body temperature begins to fall. The smooth muscles in our fingertips, in our blood vessels in our fingertips, and the capillaries in our fingertips will immediately begin to constrict to shunt the blood back to the central organs of the body to maintain body temperature there. These are automatic responses based upon what we sense. Sensory receptors are either the processed or specialized sensory neurons or cells that are monitored by sensory neurons. So there are different categories of receptors. One is called exteroreceptors. This provides information about the external environment in the form of touch, temperature, pressure sensation, and the more complex senses of sight, smell, and hearing. Proprioceptors monitor the position and movement of skeletal muscles and joints. Interoreceptors monitor the digestive, respiratory, cardiovascular, urinary, reproductive system and provide sensation of taste, deep pressure, and pain. The motor neurons, these neurons carry instructions from the central nervous system to the peripheral effectors in the peripheral tissue organs and organ systems. And our body is about has about 500,000 of these motor neurons. Axons traveling away from the central nervous system are basically called efferent fibers, and we've already discussed efferent and afferent. Two major efferent systems. This is the somatic nervous system and the autonomic or visceral nervous system. So somatic nervous system and visceral motor system. Now, as you notice, as we talk about the nervous system, there are always divisions. So don't get your divisions confused. Now, right now, we're talking about the efferent system. And again, it's composed of somatic nervous system, SNS. And I don't use the initials on the exam at all, and I won't refer to the initials here now. But last semester, I noticed several people got sympathetic nervous system confused with somatic nervous system. So, and remember, sympathetic is an antagonistic system to the parasympathetic nervous system. This is different than what we're talking about with the somatic nervous system. So, somatic nervous system and autonomic nervous system are the two major divisions of the efferent system. Somatic nervous system includes all the somatic motor neurons that innervate skeletal muscles. This is typically under voluntary control, unless, of course, we're talking about the reflexes. You touch the hot stove again and you jerk your hand back. So in the somatic system, because it's innervating skeletal muscles, this is typically under voluntary control. We have conscious control of the activities of the somatic nervous system. Now, the autonomic nervous system, on the other hand, you do not. You don't have conscious control of the autonomic nervous system. Autonomic nervous system has two divisions, sympathetic and parasympathetic system. Visceral motor neurons innervate all of the peripheral effectors other than the skeletal muscles. The autonomic nervous system innervates smooth muscle, cardiac muscle, glands, and adipose tissue throughout the body. The axons of visceral motor neurons in the central nervous system innervate a second set of visceral motor neurons in the peripheral autonomic ganglia. And ganglia is just a conglomeration, a significant number of nerves packed into one area. There are interneurons. One or more interneurons is situated between sensory neurons and motor neurons. So it's a nerve that's sitting between sensory neurons and motor neurons. More complex responses to stimuli, the greater the number of interneurons that are involved. Interneurons are involved in all higher level functioning, such as memory, planning, learning. It's these inner neurons that get dramatically affected by disease processes such as dementia. Dementia, one of the criteria for dementia is memory loss. But you also have to have one of these other higher level functionings disturbed as well. The ability to plan, the ability to be able to speak or learn also gets affected in processes such as dementia. There are multiple types of dementia: Parkinson's dementia, Alzheimer's dementia, vascular dementias, where the circulation to various areas of the brain have become compromised, and it affects these interneuron communications. When we talk about organization of neural tissue in the central nervous system differs from the organization of tissue in the peripheral nervous system. This is primarily due to the different populations of cells that are there. And the central nervous system has a greater variety of the neuroglia than does the peripheral nervous system. We also see the description, the tissue type, the histological pattern that's available there is different. We have to afford this to techniques that have been developed to be able to identify and evaluate the types of cells. This has evolved over years, and to the credit of anatomists and pathologists over time. So the neuroglia of the central nervous system, what are the cell types that are there? Well, in the central nervous system, we see ependymal cells, we see astrocytes, we see oligodendrocytes, and we also see microglia. Your book gives an illustration that goes through the different types of cells that we see in the system. To the cell types, there are satellite cells. These are little cells that actually surround neurons and cell bodies of the ganglia. We also see specialized cells referred to as Schwann cells in the peripheral nervous system, where we'll see these types of cells will basically surround the nervous tissue. They surround the axons of the peripheral nervous system. They myelinate, are responsible for myelination of the peripheral nerves. This is basically, they coat the nerve, they insulate the nerve so it helps signals to move faster, just like an insulated wire at your home. It helps the electrical impulses to stay in the wire and move a lot faster. The same impulses are a part of the nervous system as well, so myelinated system. But in the central nervous system, you don't have Schwann cells, you have oligodendrocytes. Now that's a test question. Knowing that myelinated cells in the central nervous system, the myelination is provided by oligodendrocytes in the central nervous system. And in Schwann cells, that insulation or that myelination is provided by Schwann cells. So two different, carries out basically the same function to insulate and coat the nerve, but we see it different in the peripheral nervous system versus the central nervous system. Then you also have astrocytes, which astrocytes help maintain a structure that's referred to as the blood-brain barrier. The blood-brain barrier is important because it provides not only structural support, but it also helps to regulate the movement of certain ions and nutrients in and out of the cell. It also absorbs, recycles neurotransmitters, forms scar tissues after injury. It works as a filter, so certain agents, certain toxins that may affect the rest of the body, the brain is protected from those types of substances due to the blood-brain barrier. And again, that's provided primarily by the astrocytes. And again, that's a test question as well. Microglia remove cellular debris and waste, pathogens, and by phagocytosis. That's another test question as well. So we see different types of cells. Again, satellite cells, not so important. Schwann cells important because they myelinate, but they myelinate nerves in the peripheral nervous system. Oligodendrocytes myelinate in the central nervous system. Myelin helps these nerve impulses, electrical impulses, move readily fast. Astrocytes, important cell because it helps establish the blood-brain barrier. And then microglia are like the scavengers, the phagocytes inside our cells. Microglia, they remove waste and debris. So you need to know those four different cell types, where they're located at. As you can see, 3 of the 4 are located in the central nervous system, and 1 is located in the peripheral nervous system, and their function. And finally, the ependymal cells. Now these cells are important. They're also in the central nervous system, but these cells produce the fluid that's inside of the brain. Inside the ventricle of the brain, there are tissue. These cells line the ventricles. The ventricles are the large openings inside the brain. These cavity, these spaces inside the brain are lined with these ependymal cells. They assist in producing and circulating and monitoring the cerebral spinal fluid, ependymal cells. These are located inside the central nervous system. So, I'm sorry, there is a fifth cell type. That's a test question as well, the cells that help produce cerebral spinal fluid, ependymal cells, located in the central nervous system. Now, which brings up the ventricles. Probably should have put the ventricles before the cell type. In the brain, the passageways form in large chambers inside the brain, referred to as ventricles. Central canal of the ventricles are lined with cellular layers of epithelial cells. Those epithelial cells, as I just alluded to, are the ependymal cells. This is an illustration out of your book that shows the ventricle system of the brain. As you can see, there are two large ventricles that match each other. The brain is symmetrical, and so you see a ventricle on the right. You also should see a ventricle on the left. You also see a third ventricle which runs right down the midline, and then you see a fourth ventricle as well that is in the inferior aspect of the brain and runs closest to the brain stem. Now, one reason why, when you look at a CAT scan is a slice of the brain. If you take a tomato and you slice the tomato and you look at the slices of the tomato, this is what you're looking at with the brain. One key point that I brought up before, or that I wanted to bring up now, and I don't think I talked about this before in this class. I get a little confused. It's a lot of classes, okay? But one of the things I want to bring up is when you look at a CAT scan, look at the brain and appreciate the brain. What you see on the right side, you see on the left side. So when we're reading a CAT scan, if we want to find out if a person has an abnormality, we have two sides to look at. We have a comparison. So if a person has a left CVA, we can look at the right side. If there's a change in the architecture, at a minimum, we know something abnormal has gone on in the brain. And the science of reading CAT scans is a little more complicated than that, trust me. But we have a carbon copy. We have a mirror copy to look at when we're looking at a CAT scan, because if one side has an abnormality and the other side doesn't, we can easily tell if something is different or not. So the brain is symmetrical, and it helps us to be able to diagnose acute problems that occur. But this is the ventricular system inside the body. Ependymal cells, as I already alluded to, the fluid-filled chambers of the ventricle. Fluid has to be produced by certain cells. This fluid is produced by the ependymal cells. The thickness of the wall or diameter of the passageway varies from one region to the other. Narrow passageway of the spinal cord is called a central canal. Cerebral spinal fluid. Not only is the cerebral spinal fluid located in these chambers and produced there, but it also bathes the brain. The brain actually sits in cerebral spinal fluid. That cerebral spinal fluid also goes completely through the spinal, from the brain through the brain stem all the way down the spinal cord and bathes the spinal cord itself. So, central canal, the ventricles are lined with cellular layers of epithelial cells. They're called ependymal cells that are filled with cerebral spinal fluid. This fluid also surrounds the brain, the spinal cord, and provides a protective cushion that transports dissolved gases, nutrients, waste, and other materials. You can see the relationship of the ependymal cells here in the cavity as they line the area here. You can see the meshwork or network here. The oligodendrocytes are located here. When you see an oligodendrocyte, you know you're in the central nervous system. No question about it. You also see the astrocytes here. You can see why the astrocytes are so important in maintaining the integrity of the system. Structurally, these cells hold everything into place. You see the importance of that. Now, notice the myelin processes here, the oligodendrocyte. This is the axon. It's coated, just as you would see an electrical wire coat a copper wire. The insulation allows the electrical impulses to pass very quickly along the axons. Now, there are some demyelinating diseases and we'll talk about those in a bit. There are some demyelinating diseases that destroy the myelin sheets here. As you can imagine, if you destroy the myelin, it's like taking the insulation off the electrical wires at home. And what happens? The implement that's being powered by the electricity will short out because the wires have to have a coating. And so if this coating is removed, the same thing will happen with in the nervous tissue here. The nerve will no longer be able to promulgate an action potential, and whatever muscle that nerve was innervating or whatever structure that muscle was innervating no longer functions appropriately. And there are a number of disease processes. Lou Gehrig's disease, for example, is a demyelinating disease that destroys the myelin in the central and peripheral nervous system, and thus those muscular structures that were controlled by those nerves no longer will function appropriately. So getting back to the ependymal cells, again, it has slender processes that branch extensively and make direct contact with the neuroglia surrounding nervous tissue. Functions of these connections are not known to detail. During early embryonic development, stem cells line the central canal, and the ventricles and divisions of the stem cells give rise to neurons and all of the central nervous system neuroglia, including microglia. Evidence also indicates that ependyma in the adults contain stem cells that will divide and produce additional neurons. The specific regulatory mechanism is now being investigated. Obviously, if we can identify the stem cells inside the brain, then there are a number of neurological diseases that can be eradicated if we can learn how to manipulate stem cells to differentiate into different types of nervous tissue. Now, there's certainly there's a lot of research, but at this moment we don't have the knowledge to be able to manipulate those cells like that. If we could also control this process, the treatment of strokes and several other debilitating nervous system diseases could also be revolutionized. Astrocytes. Astrocytes are the largest and most numerous neuroglia in the central nervous system. Brings up a really great movie. Did you ever see the movie Deep Blue Sea, where they were manipulating the shark's brain to produce stem cells? Did you ever see that? Was that it? Wasn't it Deep Blue Sea? Oh, that was a great movie. You haven't seen it? You need to rent that movie. Oh, that's a great movie, man. But anyway, the scientists manipulated the shark's brain, but they had to make the shark bigger because they needed a bigger brain. And so they genetically manipulated the shark, which, must be a little bit in the future. But they genetically mutated the shark so it became a smarter shark. So these were smart sharks that were. You need to rent the movie. I don't want to give away the plot. Those of you who saw the movie, I'm sure appreciated it. But anyway, astrocytes. As I drifted off. Astrocytes are the largest, most numerous cell, neuroglia cell in the central nervous system. Astrocytes have a variety of functions. Many of them are poorly understood. And again, back to the same illustration, and you can see the astrocytes. Certainly structural networking here. Also, to a degree, you can see smaller myelinated processes that are provided by these astrocytes as well. They very much help in maintaining the blood-brain barrier. Compounds dissolved in circulating blood do not have free access to the interstitial fluid of the central nervous system. Extremely important because the substances, toxins, bacterial infections that are inside the body don't get access to the brain. The brain gets protected and preserved. Now, not all. Certainly there are some substances that can get through the blood-brain barrier, but this provides a significant degree of protection for the central nervous system. Neural tissue must be physically and biochemically isolated from general circulation because hormones and other chemicals in the blood can alter neural function. The endothelial cells lining the central nervous system control the chemical exchange between blood and interstitial fluid. These cells create a blood-brain barrier that isolates the central nervous system from the general circulation. Astrocytes are packed with microfilaments that extend across the breadth of the cell, and this process, this extensive cytoskeleton reinforcement assists the astrocytes in providing a structural framework for the neurons in the brain and the spinal cord. And again, the illustration really just kind of shows you that without these structural cells, which loosely defined the cytoskeleton inside the nervous system, you would see it would lose a lot of structural support from inside the central nervous system. So very important cells. In the central nervous system, damaged neural tissue seldom regains normal function. So you have to have astrocytes moving into the site of injury, which makes structural repairs and then stabilizes the tissue and prevents further injury. Astrocytes in embryonic brain appear to be involved in directing both growth and interconnections of developing neurons. We also see astrocytes, although much remains to be learned about them physiologically, there's evidence that certainly indicates that astrocytes adjust the composition of the interstitial fluids by four main processes. And these are the four processes. They regulate the concentrations of sodium ions, potassium ions, and carbon dioxide. They also provide a rapid transit system for the transport of nutrients, ions, and dissolved gases between capillaries and neurons. We see that we see them in controlling the volume of blood flow through capillaries and absorbing and recycling some of the neurotransmitters, releasing chemicals that enhance and suppress communication across the synaptic terminals. The oligodendrocytes we've already talked about to a small degree, because these are the processes that divide the coating around the nervous tissue, the insulation. They are cylindrical processes with significant cytoplasmic extensions. Process of oligodendrocytes generally are to contact the exposed surfaces of the neuron. The function of the process is ending at the neuron, the body, yet to be determined. So much about the nervous system is yet to be determined. But we do know that many of the axons in the central nervous system are completely sheeted by these oligodendrocytes, which will basically insulate them from contact with the extracellular fluid. And again, this is in the central nervous system. Remember, in the peripheral nervous system, the Schwann cells take the same role. Axonema expands to form an enormous membranous pad, and the cytoplasmic becomes very thin. The flattened pancake, the flattened pancake type cell, it somehow gets wound around the axon, forming concentric layers in the cell membrane. Myelin sheath, this is a membranous wrapping of insulation. It increases the speed by which action potentials can travel across an axon. Again, remember, if you can make the comparison between the axon and an open copper electrical wire, that wire is much more efficient if you can insulate it. And so the brain, nervous system tissue works the same way. Many oligodendrocytes cooperate in the formation of the myelin sheath around the length of the axon. The axon is then said to be myelinated. Each oligodendrocyte myelinates segments of several axons. You can see that with the processes here. You know, the oligodendrocyte, as you can see, has several arms that reach out, and so it may myelinate more than one axon. The same cell may, I guess what I'm saying, the same cell can myelinate more than one axon at any given time. Internodes, this is a relatively large area of axon that wrapped in myelin. They're called internodes. Internodes typically range from 1 to 2 millimeters in length. Small gaps of few micrometers will separate the adjacent internodes. The nodes of Ranvier, these are gaps. They're called nodes that we'll see in different structures. Let's see if I got a picture coming up. I thought I did. Go back. The nodes, these little internodes, are what you see here, the little gaps between. You see it's not a continuous band of myelin. It's a gap. Another oligodendrocyte will provide a myelin sheet at another point, so there'll be little internodes covered up there a little bit. I don't have a nice little stretch to show you, but a node, internode here, node here. So there are these little gaps that exist there. Nodes of Ranvier. In dissection, myelinated axons appear glossy white, primarily because of the lipid content that's present. Unmyelinated axon regions dominated by myelinated axons constitute the white matter. Not all the axons are myelinated, and the unmyelinated axons may not be completely covered by the processes of the neuroglia. Such axons are common where we see relatively short axons and collaterals because the electrical impulses don't have to travel great distances. We see collaterals form synapses at densely packed neuron cell bodies. These areas are a dusky gray color, again because of the absence of the myelin. We don't see the lipid content as we saw with myelinated structures, and so this is referred to as the gray matter. Microglia, the least numerous and small neuroglia in the central nervous system are the microglia. These are slender cytoplasmic processes that have many fine branches. These cells are capable of migrating through neural tissue. Now, inside the brain, tumors can occur. Tumors are large tissue growths that can occur in the brain, the spinal cord, associated membranes. It rates approximately 90,000, probably over 100,000 deaths in the United States every year. Tumors that originate in the central nervous system are referred to as primary central nervous system tumors. You have to distinguish them from secondary tumors. Tumors are cancers that may develop in other parts of the body, can spread or metastasize to the brain, and thus that would be a secondary tumor. Secondary CNS tumors, they arise from metastatic processes that originate elsewhere. Roughly 75% of CNS tumors are primary tumors. In adults, central nervous system tumors from divisions of abnormal neuroglia rather than from divisions of abnormal neurons, because typical neurons in an adult cell don't divide. As you recall from AMP1, they require centrioles, and most adult neurons do not have centrioles, and so cell division does not occur. Central nervous system tumors central nervous system tumors in children occur when we see divisions of stem cells and neurons increase a number until children reach about the age of 4. So as a result, primary CNS tumors involving abnormal neurons can occur in children. The other thing to recognize, too, CNS tumors in children are much more responsive to therapy because children still have stem cells in the CNS, and so if these tumors are treated early and aggressively enough, stem cells are still there to be able to differentiate into nervous tissue. Primary central nervous system tumors vary in their location, thus they will vary in the signs and symptoms we see from them. Treatment may involve surgery, radiation therapy, or a combination of those therein. Addressing the symptoms just a little bit more, depending on the location of the tumor is where you'd see signs and symptoms. Tumors that occur, say, that compress, remember, the nerves, the cranial nerves from the brain, these are nerves, major nerves that come directly from the brain and innervate particular structures. The oculomotor nerve, the abducens nerve, the vagus nerve, hypoglossal nerve, and so different or various portions of the body, particularly in the head and neck, are innervated by cranial nerves. So if you put pressure on a given nerve, if you put pressure on the oculomotor nerve and the person begins to have iotosis, or if you put pressure on the optic nerve, the person begins to have a dilated pupil. It gives you an indication that the person must have an abnormal lesion and or a tumor in a particular area that's putting pressure on a particular nerve. If we know where the nerves leave the brain and we know what nerve or what tissue that nerve is to innervate, and we see an abnormality in that particular nerve, gives us an idea of what a primary tumor is. Now remember, in the brain, in brain tissue, this is a closed cavity. So the skull itself does not expand. And so when a tumor grows, it will begin to shift the brain in one direction or another. But then there's a point of a maximal expansion that the brain has, and then the brain begins to push down. So a tumor can only grow so much before it starts to affect the brain in a very severe way. When the brain pushes down, and as you study the laboratory portion of the course, you'll see the nerves that come off of the brainstem. Optic nerve being one of those, which is why we can see dilatation of a pupil when that nerve is being pressed on. But you can also have significant compromise in the respiratory centers as these tumors start to grow and push down. But they also shift the parenchyma of the brain from one side to the other. So large tumors can cause major problems because there's no room for expansion. GI tumors, you get a tumor inside your bowel, the tumor can grow huge over prolonged periods of time before you begin to notice signs and symptoms related to the size of the tumor. One of the reasons why ovarian cancer has such a high mortality rate in women is because a tumor in the ovary, there's space there, and so those tumors can grow pretty large, have an opportunity to metastasize before you can identify the cancer. Tumors inside the brain don't have to grow very big for you to start to see signs and symptoms related to it. Schwann cells are neuroliminocytes. These are from the sheath of the peripheral axons. These cells, wherever we see Schwann cells that cover an axon, the outer surface of the Schwann cell is called a neurolema. Most of the axons in the peripheral nervous system are myelinated cells, shielded from contact with the interstitial fluid, the Schwann cells, whereas oligodendrocytes in the central nervous system may be myelinated portion of several adjacent axons. A Schwann cell can myelinate only one segment of a single axon. So again, think about the oligodendrocytes. You saw many arms coming off the oligodendrocytes. They could wrap or provide a myelinated process around more than one axon at any given time. You don't see that with the Schwann cell. They wrap one segment of one axon at any one time. Illustration from your textbook, again showing you the myelinated processes here, the nodes, the inner nodes here that you see on the cells. And again, if you remember in comparison, the oligodendrocytes, that was in the central nervous system. They had arm processes that would provide maybe a myelination here, maybe one here, but then would stretch out and myelinate another nerve as well. Schwann cells are all lined up, providing the same function, but just in a little different way. Schwann cells, myelinated axon showing organizational Schwann cells along the length of the axon. Shown are stages of formations of myelin sheets of a single Schwann cell, which myelinates a portion of a single axon. A single Schwann cell can enfold a group of unmyelinated axons. A series of Schwann cells is required to cover the axon along the entire length. Most of the peripheral nervous system axons, myelinated or unmyelinated, are shielded from contact with interstitial fluid by Schwann cells. Now, demyelination is a progressive destruction of the myelin sheath, both in the central nervous system and in the peripheral nervous system. The results in a loss of sensation and loss of motor control that leaves the affected region numb and or paralyzed. Demyelinating diseases, many unrelated conditions that result in the destruction of the myelin sheath can cause symptoms of demyelination. Several important demyelination disorders may be related to toxins from different types of heavy metal poisoning, diphtheria, multiple sclerosis, Guillain-Barré syndrome are all demyelinating processes. A nice illustration in your book goes through that demyelinating process and how it can compromise the ability of the axon to be able to carry out the electrical processes. Again, when we see, we see the site of injury, what occurs, the destruction not only of the Schwann cell itself, but destruction of the nervous tissue as well in some types of injuries. Now, some type of injuries may not destroy the axonal process itself. It may just destroy the myelin itself, and if that occurs, the axon is still there, but the electrical impulses typically don't react very well at all. In this particular type of injury, we can see damage to both the myelin and damage to the axon as well. As the nerve begins to regenerate again to some degree, you can see it never returns back to its original state. It will grow, but it will be less effective than it was earlier before the time of injury, if it grows back at all. Neural responses to injury or neuron response to injury in a very limited way. The cell body, the knee cell bodies will disperse as the nucleus moves away from the centralized location. The cell will increase the rate of protein synthesis. And remember, in any type of regenerative process requires protein synthesis, so we'll see an increase in protein synthesis. If the neurons recover its functional abilities, it will return to its normal appearance. The key recovery seems to be the event underway in the point of the axon. Now consider response of the neuron. Now remember, this is repair. This is not a mitotic division where we're getting a new cell. We're talking about the axonal process of a neuron. The nucleus has not been damaged, just the peripheral arm of the nerve has been damaged, okay? And so the key to recovery seems to be the type of injury that's involved here. We consider some of the responses of a neuron as to mechanical stresses, or such as pressure applied during a crushing injury, or a pressure applied that cuts off. Remember, nerves require circulation and nutrients as well, so certain types of pressure can press on those nerves and cut off the circulation, and the nerve will eventually atrophy or die. If pressure is released after an hour or two, the neuron will recover within a few weeks, but in cases where you don't see pressure being relieved relatively in a short period of time, the damage may be irreversible. More severe and prolonged pressure will produce the effects similar to those caused by cutting off distal portions of the axon. In the peripheral nervous system, Schwann cells will participate in the repair of this damage. There's a process referred to as Walleran degeneration of an axon distal to the site of the injury. Macrophages will migrate into a particular area. They will phagocytize the debris, pick up or clean up the debris. Schwann cells don't regenerate. They will proliferate to form a solid cellular cord that will follow a path of the original axon. As neurons recover, its axon grows into the site of injury, and the Schwann cells will then wrap around that axon. The growing axon continues to grow into the periphery alongside the appropriate cord of Schwann cell. The axon may eventually reestablish, keyword there is may establish its normal synaptic contracts. If it stops growing and wanders off in some new direction, what happens in most cases, the axon will not grow back in the same path as it was before. Sometimes you'll get even additional axonic processes will grow out. So when nerves get damaged, when these axonal processes get damaged, the person may never regain sensory information as it was before, or because of the number of different axonal processes that may grow out, that area may become hypersensitive in nature as well. So it depends on the growing pattern or how that nerve wanders, or how that axonal process of the nerve will wander. The growing axon is most likely to arrive at its appropriate destination because the edges of the original nerve bundle will remain in contact. The process of regeneration, it's very limited, but it can occur in the central nervous system, but the situation gets very complicated. Many more axons are likely to be involved. Astrocytes produce scar tissue. This can prevent axonal growth. So as scar tissue forms up, the axonal process can't grow back into the same position as it was before, and it may not be able to cross the damaged area. Astrocytes will release chemical substances that block regrowth of certain actions. Now, switching subjects to potentials, there are resting potentials or transmembrane potentials in cells. This is referred to as a resting potential. All neural tissue, or neural activity, I guess I should say, begins with a change in this resting potential. A typical stimulus produces a temporary localized change in the resting potential. There can be different types of potentials that can occur. Graded potential, the effect decreases the distance of the stimuli. This is called a graded potential. Graded potentials are sufficiently large, it produces an action potential in the membrane of an axon. The action potential, definition that you need to know, the action potential is an electrical impulse that is promulgated across the surface membrane, and it does not diminish as it moves away from the source. Now, does not diminish as it moves away from the source. Typically, again, the myelin sheets, the insulation that coats these axonal processes actually will dictate that. The impulse will travel along the axon. It may go through one or two synapses before it gets to a point where that action potential will carry out a specific action. The synaptic activity that produces the graded potential in the cell membrane of the postsynaptic cell. The process typically involves the release of a neurotransmitter. Again, familiar one is acetylcholine, binds to a particular receptor. Remember, we talked about the synapse, presynaptic, postsynaptic. Action potential comes down, releases acetylcholine from the presynaptic terminal, goes into the synaptic cleft. It then causes a particular reaction either at a muscle where it connects to, or it may cause the reaction, or it may carry on that promulgation if there's another nerve cell that connects with it. So the mechanism, the model mechanism is that of the neuromuscular junction, where the axon passes that action potential down to the presynaptic terminal, where it will have contact with the postsynaptic terminal of the muscle. The release of acetylcholine transmits the action potential and the muscle will contract. The response of the postsynaptic cell ultimately depends on what the stimulated receptor will do and what the stimuli influencing the cell, or what the stimuli is. The integration of stimuli at the level of the individual cell is the simplest form of information processing in the nervous system. And again, this is the information that's being passed. The action potential is being passed down. This is the synaptic activity. This is the postsynaptic cell. In this case, we're looking at another nerve cell. But again, information, graded potential, is being passed through, making the action potential, the synaptic clefts here, to carry out additional response. You change the transmembrane potential. The resting potential is the transmembrane potential in an undisturbed cell. This is a dynamic structure that is continually modified based upon the activity and the response to stimuli. Transmembrane potential is equally dynamic, rising and falling in response to temporary changes in membrane permeability. Those changes result in opening and closing of specific membrane channels. You can have passive channels or leak channels that always stay open. And this is important because, again, typically the use of the neurotransmitter is what will stimulate the opening and closing of certain channels. Passive channels or leak channels are always open. Their permeability can vary from moment to moment depending on the nature of the channel and the response to conditions or the action potentials that comes through. Sodium and potassium leak channels are important in establishing normal and resting potential in cells. Cell membranes can also contain active channels that are called gated channels. These channels open and close in response to specific stimuli. You can also have gated channels be in one of the three states. Either they're closed, they're open. That's pretty simple. They're closed and incapable of opening as well, or inactivated types of channels. You have three classes of gated channels that are chemically regulated. They can be voltage regulated, or they can be mechanically regulated, all of which will help carry through these action potentials. Now, the chemically regulated channels respond to neurotransmitters. Again, the example is acetylcholine. This is what occurs at the neuromuscular junction. These chemically regulated channels are the most important or most abundant on the dendrites of the cell bodies of neurons. Chemically regulated channels that open in response to the presence of acetylcholine. Gated channels regulate movement of cations and anions. These are the electrolytes that have a charge. Positively charged electrolytes are cations, negatively charged electrolytes are anions. Voltage-regulated channels respond to changes in transmembrane potential. At the normal resting potential, the channel is closed. Later we'll consider some other factors that can also affect these membrane potentials. Mechanically regulated channels open in response to distortion of the membrane. At a resting potential, most gated channels are closed. The opening of gate channels alter the rate of ion movement across the cell membrane and thus change the transmembrane potential. The distribution of membrane channels vary from one region of the cell membrane or another, affecting how and where cells respond to specific stimuli. This table is out of your book 12.2 on graded channels. Tells you how graded channels, whether depolarizing or hyperpolarizing, the characteristics that are a part of these channels. Transmembrane potential, how it's affected, passively effects of the spread of action potential, graded changes in the membrane potential, and then the types of stimuli. The stronger the stimulus, the greater the change in transmembrane potential, thus the larger area would then be affected. You also need to appreciate repolarization and depolarization. The arrival or spreading out of additional positive charges shifts the transmembrane potential. Any shift in the resting potential is called depolarization. A term applies to a change in the potential from a smaller negative value as well as membrane potential, whereas repolarization, the process of restoring the normal resting potential after depolarization, this is referred to as repolarization. Now, depolarization and repolarization in response to a stimuli or the removal of a stimuli that opens chemically regulated channels such as sodium channels. You can have a state of hyperpolarization in response to the application of stimuli that opens chemically regulated potassium channels. In repolarization, when the stimulus is removed, the membrane potential will return back to its resting level. Repolarization typically involves a combination of ion movements through membrane channels that are actively pump in and out certain ions, especially sodium and potassium exchange pump. Sodium-potassium exchange pump is very important in maintaining the electrolyte balance and reestablishing the resting potential at these points where transmembrane potential is established. In the case of hyperpolarization, when we look at gated potassium channels, they have the opposite effect. Transmembrane potentials, as opening of the gated sodium channels, the rate by which the potassium outflow would increase, and then as the interior of the cell loses these positive charges, the loss of positive charges produces what's referred to as hyperpolarization, or an increase in the negativity of the resting potential from one side of the transmembrane potential to another. There are certain states of hyperpolarization at which action potentials cannot occur. There is a refractory period as well. Now, the refractory period is the time an action potential begins until the normal resting potential has been stabilized. The membrane will not respond normally with additional stimulation by the nerve. So during the refractory phase, I don't care how much stimulation continues to come in, until that normal resting potential becomes stabilized again, the membrane will not respond normally to additional depolarization or additional stimulation of the nerve. In the heart, when we look at the electrical activity of the heart, when a stimuli, electrical stimuli reaches the heart, there is an action potential that occurs. We see a definite response. There is depolarization that occurs. We see a large spike on the EKG. You've seen the spike probably on TV of the EKG. And then when we studied this in the cardiac system, we looked at the depolarization of the ventricle, which is a large electrical pattern that we see on an EKG. But then there's a little smaller resting potential that also occurs as well. It's referred to as a period of time where repolarization occurs. This is a refractory period. It's during that period of time that the heart cannot take another action potential into that area, or it can throw the heart into an abnormal rhythm. So the heart has to repolarize and has to go through a refractory period. Yes, ma'am?
Can an athlete train himself to...
Here. Now, so the sodium-potassium pump, one other concept, the sodium-potassium pump, important part of reestablishing these transmembrane potentials. Depolarization results in an influx of repolarization, which involves a loss of what's referred to as overtime, or additional electrolytes have been passed in and out of the cell. The sodium-potassium pump exchanges and returns intracellular and extracellular concentrations of ions to pre-stimulation levels. We'll see the sodium-potassium pump requires energy in the form of ATP. So we'll see ATP packed into these areas where stimulation takes place. Each time the pump exchanges 2 extracellular potassium ions, and don't focus on the numbers, just want you to appreciate what's going on here. We see 2 extracellular potassium ions for 3 intracellular sodium ions. 1 molecule of ATP is broken down into ADP. The transmembrane potential for the exchange pump is called sodium potassium ATPase, which is an enzyme because it provides energy to the pump, which will pump ions by splitting phosphate groups from ATP to form ADP. If the cell runs out of ATP, or if the sodium potassium ATPase is inactivated by metabolic poisoning, then neurons will lose their ability to function. This is a table out of your textbook, table 12.3, which shows depolarization and repolarization. Similar to, a bit different from, but similar to what we saw in EKG, where we see the depolarization of the ventricle, and then as the heart repolarizes, the transmembrane potential changes, we see a repolarization occur. And so you can go through each step, and it tells you what the electrolytes are doing. Now, as you study this and review this, I'm not going to ask you minutiae. I don't need to know how many ions of potassiums are changed for sodium. I just want you to appreciate the concept of the action potential. What occurs during depolarization, why that's important, what occurs during repolarization, and why that refractory period is there that will allow the cells to return back to their transmembrane potential, and then they're ready for another action potential. The other thing that you have to appreciate is, remember, we have a dynamic system. This is an ongoing process that happens 24 hours a day, 7 days a week inside our body to be able to function. And the last concept that I want to touch upon today is neurotransmitters and neuromodulators. Neurotransmitters are compounds that direct the effect of membrane potential and exert their effect on opening and closing these ion gates. Now, again, the model neurotransmitter that we talk about is acetylcholine, but there are other compounds in the body as well that will also carry out this response. Neurotransmitters and neuromodulators fall into one of 3 categories. You do need to know the categories. Compounds that have a direct effect on membrane potential, compounds that have an indirect effect on membrane potential, and then lipid-soluble gases that exert their effects inside the cells. Neurotransmitters, examples, and you should be familiar with each of these examples of neurotransmitters. Epinephrine, norepinephrine, dopamine, serotonin, histamines, GABA, which we'll talk about in just a bit, as well as other neuromodulators that bind to receptors in the cell. So you should be familiar with those major neurotransmitters. In these instances, the link between the first messenger and the second messenger involves the G protein that we talked about when we were discussing the endocrine system, an enzyme complex coupled to a membrane receptor. As you can see on the illustration here in the channels, you can see the effect of the neurotransmitter as it gets into the binding site, opening up channels which will allow electrons to pass across that membrane potential. Now remember, the attraction here is going to be negatively charged ions would be lined up here. When the gates open up, there's an attraction by the positively charged ions, and they'll pass through. There will be some separate gates that will allow these negative charges to pass out of the cells as well. But this is direct effect. Acetylcholine has effect on gated channels, opening up the channels, allowing those electrolytes to pass through. G protein refers to the fact that there are proteins that bind GTP. This is a high energy compound similar to ATP. Several types of G proteins. Each type includes enzymes that are turned on when extracellular compounds bind to particular receptors. This is, again, neurotransmitter, and you can see the process. It's not just as simple as a neurotransmitter binding to the receptor, but there is a whole series of reactions that take place where the G protein stimulates the enzyme amylase cyclase. ATP is required for energy. Remember, gate doesn't open up on its own. There's energy that's involved in this process which will open up the channels. The postsynaptic potentials are graded potentials developed in postsynaptic membrane in response to neurotransmitters. Two major types of postsynaptic potentials develop at a neuron-to-neuron site or synapse. That is the excitatory postsynaptic potential and the inhibitory postsynaptic potential. The excitatory postsynaptic potential, or EPSP, is a graded depolarization caused by the arrival of neurotransmitters at the postsynaptic membrane, and the excitatory potential, or EPSP, results from the opening of chemically regulated membrane channels that lead to depolarization of the cell membrane. In the inhibitory postsynaptic potential, we see the inhibitory postsynaptic potential, or the IPSP, is graded hyperpolarization of postsynaptic membrane. For example, inhibitory postsynaptic potentials may result from the opening of chemically regulated potassium channels. Inhibitory postsynaptic potentials, while hyperpolarization continues, the neuron is said to be inhibited because it's larger than what's the usual depolarizing stimulus, which is provided to bring the membrane to threshold. IPSP, this stimulus is sufficient to switch the transmembrane potential, which would normally produce the action potential, and the transmembrane potential was reset by the inhibitory potential. The same stimulus would depolarize only below the threshold that was set up before. There is a process referred to as summation, where we get excitatory potentials which will combine through a process of summation. This integrates the effect of graded potentials that will affect a portion of the cell membrane. These graded potentials may be excitatory or inhibitory or both in nature, but summation, an individual excitatory potential, has a small effect on the transmembrane potential of a typical EPSP, which produces a depolarization at a lower voltage at the postsynaptic membrane. So the forms of summation, there are two types of summations, a temporal summation and a spatial summation. The temporal summation is the addition of stimuli occurring in rapid succession. Temporal summation occurs at a single synapse when it's actively repeated. Temporal summation occurs at a membrane that receives a depolarizing stimulus from a main source in rapid succession. The effect of this secondary stimulus are added to those of the first. And that's an example of where first stimulus, second stimulus will occur. And again, action potential is promulgated, but it's one response right after the other. And spatial summation occurs when the sources of the stimulus arise simultaneously, but it arises from different locations. Innervation may be coming from more than one axon. And so you got two stimuli that are coming into the nerve, two different points, both coming at the same time. You